I'm Vincent Muser. I'm from Switzerland. I'm presently the Canada Excellence Research Chair in Genomic Medicine at McGill, and I'm very happy to have this opportunity to present our research program here. The reason I think I'm so happy to present this research program is because there is here an opportunity to change the way medicines are being discovered and developed, and there is a very broad opportunity as well for incorporating AI as a way to explore the data that are being generated and make sense out of them. So what I will do here is first share my screen, and my suggestion here is that I will present this program. It will take me probably 15 minutes, and then I will stop and pause and leave you the chance maybe to ask questions by chat. So I will not read the chat during my presentation. My presentation is entitled Large-Scale Genomics to Support Precision Pharma R&D. This slide illustrates the various steps which lead to a new drug. If we're going from left to right, we're starting with a disease, say coronary artery disease. We need to identify a target, a molecule we want to modify the functional activity or quantity, say HMG-CoA reductase. Then we need to validate this target. That means bring data which will support prosecution of this target and tell us it's worth investing. And then once target validation has been sufficiently strong, then industry is starting the high throughput screens to identify what we would call hit, which get refined to candidate. And candidates are then tested first time into man. This is mostly to test the safety and tolerability of new molecules. And once safety and tolerability have been demonstrated in healthy volunteers, phase 1, then molecules are getting tested in proof of concept studies, i.e. showing that the molecules is doing what it's expected to do. If we're taking HMG-CoA reductase, we're talking about lowering LDL cholesterol in patients with high levels of LDL cholesterol. I'll get back to that. And once proof of concept or POC has been achieved, normally drug discovery is finished and the molecule are handed into the hands of the drug developers. That means the guys who are running big phase 2b, phase 3 trials until 2 independent trials demonstrate efficacy, which normally is needed for molecules to get approval by regulatory agencies and go into the market for phase 4 or post-marketing surveillance, lifecycle management. We all know that it's very costly, it's very risky, and it's very hard to get new molecules. It takes 10 years or more, usually more than $1 billion. This morning, Joshua was telling about $3 billion. I think he's probably right. And success rate is extremely small. And so any piece of data which would help people make decisions on whether or not they want to go from one step to another at each of these decision points, any piece of data is appreciated to reduce the risk, or in other words, increase the probability of success, and also, and that's important as well, for attrition of poor molecules. Genetics and genomics have been shown to provide valuable data for a few drugs. And I would like to illustrate this point with examples. I will start with first examples showing that genetics has been really valuable for disease understanding, target identification, and target validation. And probably the best example that everybody quotes, but I think is right, is PCSK9 inhibitors. There is an interesting story behind PCSK9. PCSK9 was discovered and identified by Naïd Saïdé at the University of Montreal. He demonstrated that gain of function of PCSK9 was responsible for hyperlipidemia in certain families. But probably most of the credits are going, whether it's right or not, I don't know, to Helen Hobbs on this picture. I know Helen Hobbs well. She was my mentor. I trained for 3 years in the lab and actually replaced Jonathan Cohen as a postdoc in the lab. So what Helen has done is sequenced PCSK9 gene in a number of people, including people with very low levels of LDL. And she found an individual, N equal one individual with two separate mutations. So two PCSK9 genes were abolished. She was complete knockout for PCSK9. And she had zero level of LDL. And that was important. She had no other conditions, no heart, brain, eye, or liver condition. And that was telling industry that if you were to have a molecule to specifically lower PCSK9 and inhibit this enzyme, you have little safety issues to anticipate. It will be safe. And that's exactly what did happen. N equal 1. That's why, based on this type of studies, industry has decided to invest massively in genetics and genomics. I've got here this quote saying that Glaxo was investing $300 million to access the data from 23andMe, which, as you know, is a company generating genomic data for mostly, I would say, recreational reasons, but they are also now moving into really pharma. You can see GSK, Astra, J&J, Amgen investing $250 million in the UK Biobank of sequencing. This is exactly for this type of purpose. Another example where Biogenetics has been so instrumental is proof of concept study. And here I want to use this example again of statins. The reason I'm saying statins is that Mike Brown and Joe Goldstein got their Nobel Prize for identification of the LDL receptor and mutations responsible for familial hypercholesterolemia. So that really validated statins as target. But an important paper was published 30 years ago, 40 years ago now. That's proof of concept that statins work. And this proof of concept was demonstrated on 7 individuals, 7 patients who were defective in the LDL receptor, half of them heterozygous, and the molecule fixed the phenotype. That shows the power of human knockout for proof of concept trials. A third example where Biogenetics has been instrumental in drug discovery and development is pharmacogenetics. I'm taking here probably the most emblematic example of pharmacogenetics. This person here has developed a hypersensitivity reaction to abacavir, an HIV drug. And it is now well known that there is a genetic susceptibility to this reaction. It's called HLA-B5701. And now it's common practice to one before prescribing abacavir, because now we know that these people have a high risk and drugs shall not be given to these individuals. And so I want to summarize here the first part of my presentation. This is an overview of the examples where Biogenetics has been really instrumental for drug discovery and development. And going from left to right again, statins, Brown Goldstein's story, PCSK9, Helen Hobbs, Nahib's story, CCR5 inhibitors, enzyme replacement therapies, CFTR modulators for cystic fibrosis, and here a very well-known story as well, ApoC3 antisense oligonucleotide for a rare form of familial hypertriglyceridemia. Statins, CFTR modulators, and enzyme replacement therapies here, repurposing some examples, rare, and some examples like abacavir. Essentially, what is striking, and for me that was a lesson, is that is, if not all, most of the studies so far are based on big, rare, functional, loss of function mutations. We're going back into Mendelian genetics, essentially, what we could call human knockouts. So human knockouts are very informative. This is an area which I think, pioneered when I was working for industry. And actually, I'm showing here a paper that we published a few years ago now, whereby we systematically sequenced 200 genes of interest for pharma in 14,000 people, including 6,000 people from the cohort in Lausanne, Switzerland, which I had co-created, called the colossal study. And the question we were asking is, are there human knockouts in most genes? And the answer is yes. We can find human knockouts for essentially any gene, provided you sequence a sufficiently large number of people. That was 2012 when we published that stuff in Science. 4 years later, you could see here Geisinger's study, 50,000 exomes. The exome is the coding portion of the genome. It's not the entire genome. It's just 3% of the genome. It's the part of the genome which encodes protein. And at that time, they had technology to do 50,000. And what you can see here is if you sequence a sufficiently large sample size, number of people, you find for instance, 90% of the gene, at least one person who carries a mutation, a human knockout. And that was confirmed, for instance, recently when at the UK Biobank, they sequenced 120,000 exomes, because now technology is there. You can sequence big time exomes. And when they did 120,000 exomes, they essentially confirmed what we had predicted and was originally pointed here by the Geisinger study. There are human knockouts. They are rare. It's a matter of looking for them. But once you find them, they are informative. And that is really the rationale for the CERC program, which I have initiated here and for which I've been hired at McGill. So the research program is funded by the federal government to $10 million, McGill 10 million, and other sources, 5 million for 7 years. It's here in Montreal based, and we wanted to make it province-wide because there is also a French-Canadian rare disease heritage. I hold this slide to Claude Béret, who is in the audience here. She is my senior postdoc. And essentially, she pointed out to me that rare diseases is a big issue in Quebec, that people have been doing genetics for 200 years now in this province, that there is a whole series of diseases which are known to be very specific to this population. But surprisingly enough, there is no systematic analysis of the architecture of functional coding variants, that means the human knockouts in particular, in this population. And that's really the gap that Claude and our team are wanting to fill. So the mission of the CERC we have received is shown here, is essentially to contribute to the discovery and development of innovative therapeutics using genomic medicine. Sounds very simple, it's a bit complicated. Our vision here is to build up over the 7-year period a one-stop shop to support drug discovery and development. We want to capitalize on functional variants, including these human knockouts, the predicted loss of function, PILOF. And we want to capitalize as well on the experiments by nature in the French-Canadian and cosmopolitan population of Quebec, the French-Canadian heritage being mostly due to the fact that we now have a few million people, which are originally just derived from 8,000 people who colonized this province back 300 years ago. So the strategy is shown here. That's the strategy we want to put in place now for the next years. We want to build up first a large reservoir of patients that we can sequence the genome or the exome. We call it the Quebec Clinical Mendelian, and it's really McGill-affiliated hospitals, Carthagène, which is a population-based study of 40,000 people, and our Quebec partner hospitals. And our plan is to do whole exome sequencing, like I've been showing you, or whole genome sequencing, and identify these carriers of mutations, and then ask these individuals, these carriers, to go back to the clinic, or if data is available through their medical records, and here AI would be really so important, to examine these people very carefully, and potentially, like the statin story I mentioned to you before, do pharmacological interventions on genetically selected individuals. To that end, we need a very strong computational genomics group. And so Claude is part of this group. We recently hired as well a very talented individual from Michigan University, Daniel Talion, who is now joining our team and will be heading the computational genomics group. What I want to show you next in this last part of my presentation is 3 enabling projects that we have initiated since my arrival here. And one project is to show our capacity to generate a province-wide effort. And actually, COVID, unfortunately, gave us this opportunity to demonstrate our capacity to do so. Is the Quebec Covid-19 Biobank, which I have presented before. Claude and a group have generated a first map of the coding variants in the French-Canadian population, which I will show you one example of. And we are also developing at the Genome Center technology for sequencing a large number of exomes at limited cost. So I just want to go quickly over these 3 programs. The first is the COVID-19 Biobank. I've been presenting that before this afternoon. It's a mandate which we received from the Fonds de Recherche en Santé du Québec, Carole Jabet, the scientific-in-chief of the province, Rémi Quirion, and Genome Québec, Daniel Coder, before the wave. We didn't have any patients in the hospitals almost there. And they gave me the mandate to build a biobank so that we can have the data and samples from future patients hospitalized in the province. We received the mandate in March. 2 days later, we had a task force in place. 10 days later, we got approval from the ethics committee. 11 days later, we started recruitment, and Brent Richards has been extremely instrumental there at the Jewish General Hospital. A couple of months later, we had 1,000 patients recruited, and we got the Public Health Agency of Canada to support further our projects. The first GWAS data, so the genotyping data, was released 17th of July. We have recruited so far 1,400 people. At the end of wave 1, we had 1,200 participants already, which makes it one of the largest biobanks in the world for COVID purposes. And I'm just wanting to show you here one piece of data. Again, it's from Brent Richards' lab at the Jewish. But what Brent has been doing is use genetics to identify potential drug targets for repositioning and new indication into COVID-19. And I don't have time to go into these details, but just want to illustrate here the power of genetics to identify 4 different molecules circulating in blood, which are associated potentially causally with COVID-19, and as such represent potential drug for repositioning for COVID indications. The second enabling project, as you remember, is analyze the genomic architecture of coding variants, including these human knockout variations in the French-Canadian population. And that's really the work that Claude has been doing in collaboration with John Ryu, John being at the Institut Cardiologie de Montréal. The data shown here refers to 2,500 exomes, which have been sequenced by the MIT. And what Claude has done is looked at all the mutations in these 2,500 exomes. And she found 9,000 mutations which are abolishing the function of the protein, so human knockouts. They are rare. Most of them, very vast majority, are found only once in 2,000 people. And then what she did is she looked at the relative frequency the frequency in the French Canadian population compared to the frequency in a non-Finnish European population. And so if these variants had the same prevalence in Quebec and in the European population, we would have essentially a distribution around 1. But we have here a second peak, illustrating enrichment. These variants, and look at the numbers, 1,400, for instance, here, 1,200, these variants are more than 10-fold more prevalent here than they are in Europe. And when you plot them individually, each dot here representing one variant. And this is the line of identity, Europe, French Canadians. If we had the same frequency, we would be around here, but there are lots of variants, actually, which now are known to cause diseases in our French Canadian population. And so it's now the demonstration that we have here a unique population to work with, because these populations have variants that are found almost nowhere else and can be used to support drug discovery and development uniquely. The third thing is how we can detect these rare coding variants with new technologies. We want to spend less than $200 to analyze the entire genome for our search purposes, and actually we're making real progress into this direction. So to conclude my presentation and open up the discussions, I hope I have convinced you that the large-scale genomics represent a powerful new tool in the box to support target identification, target validation, and also to open up next generation of proof of concept trials for investigation medicine. Very small trials, very informative, very decisive. We strongly believe that the environment here in Quebec is unique to capture this opportunity because of the founder population and the enrichment I mentioned to you, because of the environment, the academic, industrial environment, and all the capabilities in genomics and AI, plus the clinic. The CERC, which I'm chairing, is now in place. We have the foundations in place. We have enabling projects demonstrating a capacity to build up this program. And so we're moving towards a unique source of data, which will combine clinical, laboratory, and genomic data for AI and other explorations. Just want to finish up here my slide by expressing my gratitude to these people here, including Claude, and the people who made it possible to build up the Quebec Biobank, and John Ryu and his team as well for sharing generously with us his exome data. So I'm done, and I thank you for your attention. What I propose now is to leave 2 minutes if you have questions, and maybe you can ask your questions in the Q&A box, and I'll come back in 2 minutes to read them. Okay, I have the first question here from Muhammad. Does CERC workflow involve any transcriptomics data at any of the steps? The answer is yes, we will be looking at transcriptomic data. Transcriptomic is not our focus, but transcriptomic data can be very helpful because they allow to identify as well coding variants, of course. That's the first thing. The second thing is, in the Quebec Biobank, which I briefly described here, we are doing systematic transcriptomic analysis, and we are collecting PAX gene tubes for doing transcriptomics on peripheral white blood cells isolated from COVID-infected patients. So that will be an arm that we will be exploring. Nathaniel, can I read your question, Nathaniel? What is the framework with which CERC plans to work with companies? During or after sequencing efforts, how would data be shared? Would local and international companies be of interest? Well, that's an important series of questions, Nathaniel. Thanks for raising that. The first thing is, yes, we are developing this resource to promote the discovery and development of new drugs in Quebec and in Canada. That's the first thing. The second thing is we want to work with the network of companies which are active here. And for instance, our goal in 3-4 years, when we have put everything in place, is that companies could come to Quebec to us and ask questions. Do we have genetic data to support our target or validate it to find optimal indications or alternative indications? Do you have patients who carry mutations within our target genes? And can we do trials on these individuals? So that's the goal, to make a one-stop shop for companies. I have worked in companies. I've spent 10 years in pharma, so I'm lucky enough to have been behind the scene and have sort of real hand exposure into what it is to discover and develop new drugs. I think that these CERC program builds a unique spot which is very difficult for pharma to access. Pharma is so good at doing the, and CROs are doing the phase 1, the volunteer studies, healthy volunteer studies. They are so good at doing the large phase 3 trials. But this sweet spot here, proof of concept studies, are very difficult. And we believe that we can occupy this spot. We believe that this spot is unique as well. If we're thinking computational genomics, functional genomics, pharma can do that in-house. They have access to data, enormous amount of data. You get biobank data. They don't have these patients to do this type of studies in this sweet spot. So that's where we want to be. That's where we want to promote also interactions with industry. We want to do that in a very scientific manner, of course. We are academic, we want to stay academic, but also we are fully aware that it's not the job of academia to develop new drugs, that new drugs are being developed by industry, and we can partner with them in a very healthy and sustainable manner. Does it answer your question, Nathaniel? If not, please drop me a note. Martin, you're asking me about any plans to investigate non-coding variants. The answer is absolutely yes. We are interested in non-coding variants, but we will initially focus on coding ones for 2 reasons. The first reason is we may have some of them which are uniquely present in our population. The second reason is that they are easy to interpret. Now, we are interested in non-coding variants for a variety of reasons as well. Firstly, non-coding variants can be very helpful for polygenic risk scores. And we will use polygenic risk scores to, for instance, enrich in carriers of coding mutations. Second, there are some non-coding variants which are, of course, very active transcriptionally, for instance. And so we want to keep this capacity to identify people who have, say, gain or loss of expression of genes. To that end, what we are doing now with Claude and the group is to develop technology to do exome at a very big depth. So making sure that we identify these coding variants and the rest of the genome at a shallow depth to identify non-coding variants. We are also developing with Daniel Talion and Claude new tools for imputation of coding and non-coding variants, including as well insertion deletions. If there are no other questions, let me thank you all for attending this presentation. I'm certainly happy to answer questions. Otherwise, we have a website called cercgenomicmedicine.ca or something equivalent. And thank again for the organizers for giving me the chance to share with you our plans. Thank you. Bye-bye.